Hi, so today I'll be giving a talk on my research topic titled Small Plaque Variants of EV-A71 as Vaccine Candidate. First of all, enterovirus 71, EV-A71 is a major causative agent of the hand, foot, and mouth disease (HFND), which can be associated with severe complications such as aseptic meningitis, acute flaccid paralysis, and pulmonary edema, which could lead to fatality. EV-A71 was first isolated from a young child in 1969 in California, United States, and in the later years, sporadic outbreaks have arose, causing large outbreaks in the Asian Pacific region, including China, Malaysia, Vietnam, and Singapore. In June 1997, 4,253 cases were reported in Malaysia due to EV-A71-associated HFND outbreaks, with recurring fatalities in year 2000, 2005, and 2006. Further outbreaks took place in 2009 and 2012 with a cyclical pattern of 2 to 3 years. Among the countries in Asia, China has reported yearly HFND infections of over 2 million cases. EV-A71 was reported to account for close to 50% of the HFND infection yearly. Up to date, there is no known FDA-approved vaccine that could prevent severe EV-A71 infections. The current vaccine in use are inactivated vaccines, IVs, approved by the China FDA such as Sinovac, Beijing Vigo, and CAMS. The IVs were reported to have an average of 94.8% efficacy, but multiple booster injections are needed every 6 to 12 months, and thus there is concern with high cost and patient compliance. This is inconvenient for people who live in rural areas that need to travel all the way to cities such as Beijing and Shanghai to receive the vaccinations. EV-A71 is an RNA virus which has a low fidelity RNA-dependent RNA polymerase that can introduce 10 to the minus 3 to 10 to the minus 5 mutations per nucleotides copied in each replication. This can introduce misincorporation of 1 to 2 nucleotides in the genome. As a result, spontaneous mutations in the EV-A71 genome were frequently observed. Researchers will need to understand how the spontaneous mutations affect virulence before using the strain as a vaccine seed strain. Therefore, this poses as a challenge on the development of an EV-A71 vaccine when it comes to choosing a vaccine strain that is able to elicit broad neutralizing antibody titers against different subgenotypes in an outbreak. Any selection of a subgenotype may not be able to provide sufficient protection against other subgenotypes. The EV-A71 wild-type population comprises of several quasi-species, referring to a large RNA population with various mutants carrying genetically related genomes, hence showing plaques of various sizes. Small plaque variants were often used as live attenuated vaccine (LAV) candidates due to slower replication, leading to less spread and cell killing, which implies lower virulence. A viral plaque variant with desirable characteristics such as reduced virulence may serve as a candidate for LAVs, which can be administered orally like the polio vaccine. Live attenuated vaccines are able to elicit both humoral and cellular immunity to provide lifelong immunity. An effective orally delivered EV-A71 LAV will provide the world with more vaccination platforms against EV-A71 infection, moving us a step closer to eradicate this neurotropic pathogen that is likely to replace poliovirus after its eradication. And that's all from me. Thank you.